Hello, everyone, and welcome to today's webinar titled The Invasive Niche of the Basal Cell Carcinoma as Revealed by Integrated Multi-Omics. This webinar is part of the Cancer Research and Immuno-Oncology Virtual Week, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by NanoString Technologies. For more information about NanoString, go to nanostring.com. Now let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located on the left of your screen. You can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Now, if you are having any trouble seeing or hearing this presentation, just click on that Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credit link located in the abstract window to the left on the screen and follow the process to obtain those credits. I now present today's speaker, Laura Yerly, a PhD student at Lausanne University Hospital. For a complete biography of our speaker, please visit the Biography tab at the left of your screen. Ms. Yerly, welcome. You may now begin your presentation.
Hi everyone, I will present you today our work where we show how multi-omics integration reveals cellular and molecular interactions governing the invasive niche of basal cell carcinoma. Tumor cell invasion is a hallmark of cancer. Tumor cells invade the surrounding tissue to escape starvation, and tumor cell invasion can lead to metastasis and is associated with poor prognosis. It is known that tumor microenvironment plays an important role in tumor cell invasion. However, it is still poorly understood which cells are interacting with the tumor cells and what interaction is happening. So in order to understand a bit better these interactions, we decided to use basal cell carcinoma as a model. Basal cell carcinoma is a skin cancer. It is the most frequent human cancer worldwide, and despite genetic homogeneity through defect in the hedgehog signaling pathway, basal cell carcinoma can display different clinical and histopathological spectrum. In fact, basal cell carcinoma can be divided in different subtypes. The nodular BCC subtype, showing no infiltration and often surrounded by a basement membrane, suggesting that there is low interaction with the surrounding stromal cells. Basal cell carcinoma can also be divided in an infiltrative subtype, which form single-cell strand infiltrates without basement membrane, suggesting high interaction with the surrounding stromal cells. Furthermore, recent studies show that genes characterizing infiltrative BCCs through RNA-seq analysis or extracellular matrix remodeling genes. However, this is not black and white. There is a kind of gradient between nodular and infiltrative BCC subtypes, suggesting that tumor and stroma have transcriptional reprogramming. So our question was, can we transcriptionally characterize tumor and stromal cells composing the invasive niche? So to this end, we started by performing digital spatial profiling with the Geomics technology of NanoString on nodular and infiltrative BCCs. We took biopsies that we stained with pancytokeratin antibody and counterstained with cancer transcriptome atlas probes. Then we draw region of interest at the tumor-stroma interface. Then, based on the presence or absence of pancytokeratin staining, the region of interest were divided in two areas of interest. The tumor area of interest if the pancytokeratin staining was positive, and the stroma area of interest if the pancytokeratin staining was negative. Then the regions were UV cleaved and sequenced, and then we performed data analysis. So from the tumor area of interest, we extracted signatures. So we extracted a spatial tumor nodular signature from the tumor nodular area of interest, and we extracted a spatial tumor infiltrative signature from the infiltrative tumor area of interest. When we look at the PCA plot, we observe that the signature segregate well the different BCC subtypes. We performed the same from the stromal part, so we extracted a spatial stroma nodular signature from the nodular stroma area of interest and a spatial stroma infiltrative signature from the infiltrative stroma area of interest. When we look at the PCA plot, we see that here again, we have a good segregation between the subtype stroma area of interest. Then we have the limitation that we don't have a single cell resolution. So to counteract this limitation, we decided to perform single cell RNA sequencing on 5 infiltrative BCCs. So we enzymatically dissociated 5 fresh tumor biopsies. Then we fax sorted single living cells, performed chromium 10x single cell RNA-seq, and analyzed the data. We obtained the expected cell subtypes. We have epithelial cells, T cells, melanocytes, myeloid cells, endothelial cells, fibroblasts, cycling cells, pericytes, mast cells, and B cells. But with single cell, we don't have the spatial location of those cells. So to counteract this limitation, we decided to combine both techniques, and we applied the spatial signatures obtained from digital spatial profiling to the single cell RNA-seq data. When we applied the spatial tumor signature, it highlights the tumor cells. And when we applied the spatial stroma signature, it highlighted a particular cell types, which are the fibroblasts, suggesting that spatial signatures identifies tumor cells and fibroblasts composing tumor-stroma interface in the invasive niche. Then we wanted to characterize a bit further those two cell types. So we started with the tumor cells. We subclusterized the tumor cells and then applied the difference between tumor infiltrative and tumor nodular signature scores. And we can see that we have a region that is blue and is enriched for tumor nodular signature, and then a gradient going toward the red region that is enriched for the tumor infiltrative signature. So we decided to rank the clusters according to the signature score of the difference from lowly infiltrative to highly infiltrative. Then we characterized a bit further those tumor cells by looking for differentiation signature of keratinocytes. And we observed that we have an enrichment for basal signature in the lowly infiltrative region and for differentiation signature in the highly infiltrative clusters, suggesting that tumor cells with their gradual infiltrative phenotype have also a gradual differentiation phenotype. Then we were interested to know how those cells migrate. And to this end, we look at different migration signature. So first we look for epithelial to mesenchymal transition signature. And we observe that we have no particular enrichment for this signature. But when we applied the collective migration signature, it highlights the region where we have the highly infiltrative tumor cells, suggesting that our tumor cells show a gradual infiltrative phenotype with higher collective migration features than EMT. Then we wanted to take advantage of the pseudo-time analysis and look whether we can recapitulate our findings. So we take as starting point the low infiltrative cluster, and we observe that it illustrates the infiltrative phenotype because we have a decrease of the nodular marker genes along the pseudo-time and an increase of the infiltrative marker genes along the pseudo-time. This was confirmed by FISH. So in the lowly infiltrative region, we observe that we have a high level of the nodular marker gene and a low level of the infiltrative marker gene. In the medium infiltrative region, we have an intermediate signal of the infiltrative marker and an intermediate signal of the nodular marker. And when we go in the highly infiltrative region, we have a high expression of the infiltrative marker and an absence of the nodular one. Then we confirmed our results by looking at the basal marker genes, and we see that they are decreasing along the pseudo-time, whereas differentiation markers were increasing along the pseudo-time trajectory, as well as collective migration markers. Then we wanted to characterize the other cell types, so the fibroblast. We applied the difference between stroma infiltrative and stroma nodular signature scores, and we observe here that we have again a region that is blue enriched for stroma nodular signature, and then a gradient toward the red where we have an enrichment for the stroma infiltrative signature. So we ranked again the clusters from lowly infiltrative to highly infiltrative. Then we wanted to characterize the fibroblast further and apply a cancer-associated fibroblast signature, and we observed that both fibroblasts surrounding lowly infiltrative tumor cells and fibroblasts surrounding highly infiltrative tumor cells are enriched for this signature. However, when we look at the heat map, we observe that the genes responsible for the signature enrichment are different between the fibroblasts surrounding highly infiltrative tumors and the one surrounding lowly infiltrative tumors. So to characterize them a bit further, we decided to look at differentially expressed genes between the FC4 cluster surrounding highly infiltrative cells and the FC1 cluster surrounding lowly infiltrative cells. And we observed that we have an enrichment in the FC4 for extracellular matrix organization and extracellular structure organization genes, suggesting that the CAF found in the invasive niche harbor extracellular matrix remodeling features. So we can summarize the first part. So we showed that infiltrative tumors show a gradual infiltrative phenotype going from low to medium and highly infiltrative, that the highly infiltrative tumor cells display differentiation and collective migration features, and that the highly infiltrative tumor cells are surrounded by extracellular matrix remodeling CAFs. Then we wondered whether we can infer a crosstalk between spatially connected tumor cells and CAF. And to this end, we decided to look for the enrichment of cancer-associated fibroblast activator. And we observed that we have a particular enrichment of the NHBA gene in the highly infiltrative clusters. So NHBA is a gene encoding for activin A, and activin A is a homodimer of the inhibin subunit beta A. It is a member of the TGF-beta superfamily. Activin A is a secreted protein known as CAF activator. Furthermore, Concrema et al. Published recently that activin A promotes carcinogenesis via fibroblast reprogramming in squamous cell carcinoma, which is another kind of skin cancer. So this was interesting for us because we decided to look whether our fibroblasts were influenced by the presence of activin A. So we decided to apply the published activin A exposure signature, and we observed that the signature was particularly enriched in the FC4 cluster corresponding to the fibroblast surrounding the highly infiltrative tumor cells. So we confirmed this finding by performing FISH, and we observed that in the superficial part of the tumor, where we have lowly infiltrative tumor cells, we have a low expression of NHBA and a low expression of fibronectin-1 in the surrounding fibroblast. So fibronectin-1 is a member of the activin A exposure signature. But when we go deeper in the tumor, we have highly infiltrative tumors that show a high level of NHBA, and we have also a high expression of fibronectin-1 in the surrounding fibroblast. So with this, we can conclude that we show that infiltrative tumors have a gradual infiltrative phenotype from low to medium and highly infiltrative, that the highly infiltrative tumor cells display differentiation and collective migration features, and that highly infiltrative tumor cells are surrounded by extracellular matrix remodeling CAFs. And when we zoom in the invasive niche, we have a high expression of NHBA in the highly infiltrative tumor cells that encodes for activin A, and activin A acts on the surrounding extracellular matrix remodeling CAFs. So with this, I would like to particularly thank my PI François and my colleague Christine and all the other people who worked on this study. And I will be happy to answer to questions.
Thank you, Laura, for that informative presentation. We will now start the live Q&A portion of this webinar. And if you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located at the far left of your screen. So let's get started. It looks like we already have a couple questions coming in, Laura. Our first question: did you have any contamination between the tumor and stroma AOIs in the spatial experiment?
So yes, we had some. When we looked at the genes present in the tumor signature and in the stroma signature, some genes were overlapping. So in order to take out the contaminating genes, we looked in the single cell data, whether the gene was present, for example, a gene in the tumor signature, if it was mostly expressed in the stromal part, we removed it from the tumor signature. And also if it was equally expressed between tumor and stromal cells, we also removed it. We kept only the genes that were more expressed in the tumor cells compared to the stroma in the tumor signature. And we did the same for the stromal part.
Thank you so much. And why did we have so many immune cells in comparison to fibroblasts in the scRNA-seq experiment?
So this might be due to the dissociation protocol, because immune cells are can more easily be isolated than fibroblasts that are adherent to the matrix, and the infiltrative BCCs are known to be really fibrotic. So there is a lot of extracellular matrix. So this might be the cause why we don't have so many fibroblasts.
Thank you so much. And Laura, did you have any closing remarks for our audience today? It looks like that was the last question coming in at this time.
So just thank you to attend to this webinar and for the question.
Well, thank you, Laura Yerly, for your presentation today and your important research. And before we go, I do want to thank our audience for joining us today and for their interesting questions. And I'd like to thank our sponsors, NanoString Technologies, for sponsoring today's webinar. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speaker via the contact information you provided us at the time of registration. And this webcast can be viewed on demand for 2 years until October 4th, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who missed today's live event. Until next time, take care, everyone. Bye-bye.